Dear students, recently we studied different classes compound which we called biopolymers. They formed the structure of life, the barrier preventing us from outside. Today we will study substances which interact with biopolymers causing the physiological repulse, and we can name them, in contrary to biopolymers, small molecules. There are a lot of small molecules, some of them we discussed in previous topics. They belong to different classes of heterofunctional compounds, but today we mostly will discuss steroids. They are possessing a lot of function, and for animals and human beings, mostly of hormone activity. Uh, just repeating again that there are a lot of biologically active compounds that belong to different classes, and the main difference from the classes we studied before, they have relatively small molecular weight, usually before 500, and they interact with a protein or with some other uh, biopolymers to cause physiological repulse. I said to you that today mostly we'll study steroids, and the peristructure of steroids, yes, consists of four cycles. Just A, B, C, D. This is a standard presentation. It's not prohibited to draw the molecule another way, to flip it, to tumble, but just in such a way, some special descriptors for steroid nomenclature and stereochemistry will be available. Just we can find two bicyclic systems. Yes, the bicyclic system of decaline and hydrogen. Fused together, they form a gonane structure. Just a few words about numbering. It's rather strange. Yes, but it's historic and based on the numbering of decaline and hydrogen structure. Yes, we start from decaline, bicyclic system, and then again from hydrogen. Just we can see that gonane skeleton, when saturated, contains 1, 2, 3, 4, 5, 6 chiral center. This is the maximum number of chiral centers, so we can find a lot of stereoisomers. For sure, when when we will see some double bonds attached to chiral centers, we will lose some of stereoisomers. Just a few words I want to remind you about the stereochemistry of decaline. Yes, hydrogens can be arranged in plain a different way. Just when both hydrogen from one side, we can say about cis decaline, and when saying about chair conformation, just for cis decaline, one hydrogen will be equatorial and one hydrogen attached to this angular carbon will be axial. Cis axial position of two vicinal hydrogen corresponds to cis decaline, and vice versa. Transdecaline, um, it possesses both hydrogens in axial position, and different steroids, they are belonging to these two series, either to cis position of two hydrogens or to trans position. As for hydrogen, again, we can have two different variants. Yes, cis hydrogen and trans hydrogen for the axial position of hydrogens. Just one saying about natural steroids. Yes, When we'll say about synthetic steroid, everything is possible. But when we say about natural steroids, uh, the fusion of B C cycle is mandatory. It will be always trans. As for A, B, they can be both trans and C steroids, and just because of the subdivision, steroids belonging to trans or cis series, and C, D mostly trans. There are some exceptions, but mostly trans configuration. Just for steroid, some special approaches to stereochemistry are possible when we present steroids in standard way, A, B, C, D, from left to right. Just we can name any substituent, including hydrogen, in alpha beta descriptors. If substituent, again including hydrogen in angular positions, will be in front of the plane, we are saying about beta position. When when substituent will be behind the plane, we're saying about alpha position. Just this position, yes, it will be mandatory, mostly for methyl group in steroids, but here you see the hydrogen. So trans configuration, trans configuration will have hydrogen behind the plane, and we can say about 5 alpha steroid because 1, 2, 3, 5. And for cis configuration, yes, again, because this substituent will be in front of the plane. For cis configuration, the hydrogen in this position will be in front of the plane. We can say about 5 beta steroid. And again, I remind you that all the other substituents, for example, here hydroxy group in front of the plane will be beta hydroxy group. For example, this position behind the plane will be alpha hydroxy group. But again, all steroids can be belonging to two series. When hydrogen behind the plane, it will be trans configuration. We can say about alpha series, 5 alpha series of steroids. And when hydrogen will be in front of the plane, it corresponds to cis configuration. We can say about 5 beta series. Yes, and you need to see the correspondence between plane and check conformations. Just alpha steroids, yes, alpha steroids, they have axial hydrogen in angular position, and beta steroid equatorial hydrogen in 5 position. Just for sure, we can name steroids based on gonane parent structure or even on parahyd of cyclopentaphenanthrene, but most natural steroids can be classified in few groups based on the character of substituents in the 10, 13, and 17 position. Yes, here you see the general scheme. You see that the minimum number of substituents is typical for a strain structure. Yes, so when here we have only one methyl heme and here no substituents, we will have about ester, a strain structure. When we add one more methyl group, yes, we will see about androsten structure, and so on and so on. Then pregnane, choline, and cholestyne. Just one by one, we will consider all possible steroid structure. We'll start with the strain with the only methyl group. You can see this is 13th position. This is a strain structure. So if you will see the steroid with carbon-18 parent structure, it will be from a strain series. Just a strain is this is something from the structure, but when we add methyl group, we will obtain understrain structure to move carbons pregnane structure. Yes, and that is why it will be 18th, 19th, 20, 21st. Then three more carbons, 22nd, 23rd, 24th, Colane structure, and finally cholesterol with 27 carbons. So we discussed only the structure, the structural approach. When saying about the physical properties, steroids mostly water insoluble, but they are well soluble in non-polar solvents, and because they have a lot of current centers, uh, steroid solutions, solutions of natural steroids, non-racemic steroids, they have optical activity. Steroids, because of high lipophilicity, can pass through biological membranes, nucleus membrane, and just interacting with different biopolymers, including DNA. Just now we will discuss different subgroups of steroids one by one. Just I regard you that only one methyl group in 13th position. Yes, it will be 18th carbon correspond to a strain structure. This is something from the structural chemistry. When saying about physiological activity, estrogen-based steroids, natural estrogen-based steroids, they possess estrogen activity, estrogen, so-called female hormones. The representative of estrogens or estrogen-based steroids is estradiol. This is common. This is historic name, but I think it's easy to memorize it because it contains really 2-hydroxy group. Just it's very interesting that estradiol contains benzene ring. So in real we have no cyclohexane here, and in other cases we name benzene ring as benzene ring. But to um, to join estrogens to other steroids, we name estrogens in a regular steroidal manner. So this will be ester, ester structure. Ester, it means fully saturated, and we need to name double bonds. Nobody, nobody names benzene as cyclohexatriene. But, but